Good morning, everyone. I'd like to introduce our Grand Round speaker today, ABarnes, who's a third-year clinical pathology resident. Aaron has a long history here at the university prior to joining our residency and did a PhD in microbiology working on the biology of the Enterococcus under the tutelage of Dr. Gary Dunny, who's sitting in the front row. Thank you for coming today, Gary. Aaron did some fantastic work in Gary's lab, did 2 years of a postdoc working on Enterococcus, and then joined our residency. Aaron has been very energetic during his residency. He has other research projects that are going to be coming to fruition, including about 3 with me that we hope to be able to either launch or finish soon. And so without any further words, it's Aaron. Thank you, Aaron.
Thank you, Dr. Frey. So as Dr. Frey mentioned, I've been working on Enterococci for quite a long time, and today what I'm going to do is talk about a pair, maybe even the third one if I have time, a pair of mammalian model systems that I started working on in my postdoc. So as a PhD student, I was looking at lots and lots of in vitro work. So trying to figure out how biofilms work in Enterococci, how these things form. But it became pretty relevant pretty quickly that it was going to be really important to sort out what's actually happening in animals, because that's what we really care about, is what's happening here. And so I started working, like I said, in my postdoc, and then I've continued that work in the last couple years. And so we'll talk about that without any further ado. I have no disclosures. So my objectives today are to talk a little bit about biofilms. A lot of this work, when I presented it, it's been to other microbiologists, so I've tried to sort of refocus this, because not everybody in the room works on biofilms. So talk about it both in the disease and the normal state, because biofilms also have a role as commensal organisms in our GI tract and on our skin and other places. I want to talk specifically about the biofilm formed by Enterococcus faecalis, which is the bug that I study in these model systems, and then talk about some of the research opportunities that happen once we start to think about how biofilms work in disease. So Enterococcus faecalis is a low GC gram-positive diplococci. Up until about the mid-'80s, it was considered to be group D streptococci. Then we realized that wasn't really an appropriate term for them anymore, so we moved them over. They are normal flora in our GI tract. About 85% of us in this room have it in our GI tract. Honestly, that's probably 100%. They just tend to hide out a little bit. But they live in pretty much all animals that are terrestrial. They're in insects. They're everywhere. They do occur in fish, though, at a lower degree. There's some interesting evolutionary reasons that might be true. Enterococci as a group are really famous for 3 things. The first is that they are intrinsically resistant to 3, or depending on how you look at it, 4 antibiotic classes. This is a big deal when you get an infection with an enterococci because it gets to be hard to treat. They also have a very well described and understood horizontal gene transfer system. So they are really good at shipping both antibiotic and other genes back and forth between them. And they are extremely environmentally adaptable. Enterococci in general will grow between about pH 4 and about 9.5. They grow at 8 degrees C. They grow at 45 degrees C. They can survive 60 degrees C for 30 minutes. Those are some pretty wide extremes. I mean, they will grow in 6.5% salt. They will grow in 40% bile. I mean, this is pretty much the hardest thing to kill that doesn't form a spore. I mean, they are really, really tough bugs. They also form biofilms, and they do this really quickly. A lot of the work that I did in my PhD was to sort out how fast they do this, because the dogma used to be that biofilms form over days to weeks. Biofilms form in hours. You can start to see biofilms form in 2 to 3 hours. You get in vitro, you get pretty definitive biofilm formation after about 6 hours. Clinically, this is relevant because we can't think anymore, how long have they had this infection, has this formed a biofilm? No, it's formed a biofilm by the time you found it. It's already done that. And that's important because biofilms are really hard to treat. So once a bug of any sort, once a bacteria of any sort has gone into a biofilm state, it becomes somewhere between about 10 and 1,000-fold more resistant to antibiotics. And we can't give somebody 1,000 times the normal dose of vancomycin. That's not a thing you can do. So what are biofilms? So biofilms occur when bacteria attach to a surface and start to produce an extracellular matrix of some sort. This is highly variable. We used to think that biofilms were biofilms. That's completely not true. It depends on the bug, it depends on where it is, it depends on what it's doing, it depends on the population of other bugs that are around it. And what I mean by what is the biofilm, this can be polysaccharide-based, protein-based, DNA-based, all of the above. In some organisms like E. coli, this is very easy to sort out because they basically use two proteins compose about 85% of their biofilm. That's not true in Enterococci, nor is it true in most bugs. Mostly this is a well-formed matrix structure that exists. And again, this changes based on time, infection, what else is going on in the system. Not only does this make them biofilm resistant, this makes them basically refractory to almost anything except surgical removal. So this is why when somebody has an infected hip joint or an infected valve, the first thing you try to do is, can we get this out? Obviously, removing someone's prosthetic hip joint is not ideal, so we'd like to figure out ways around that. Also, the bacteria are communicating with each other. We've known this for years, but it becomes much more important when you realize that not only they're communicating with each other, they're taking on specialized roles inside of a biofilm. So we have cells attaching to a surface, taking on specialized roles, making an extracellular matrix. This is basically the definition of a tissue. This is what we call tissues when we talk about mammals or higher organisms. We say, a liver is not just a random assortment of hepatocytes. There's a structure, there's cells doing different things. Biofilms are basically the bacterial equivalent of a tissue. Okay, so, at least 75% of all chronic bacterial infections involve a biofilm. Again, this number is probably closer to 100%, it's just harder to prove. And this occurs in pretty much any place in your body where you either have limited access to an immune system response or a prosthetic of some sort. So this can occur in your sinuses, this can occur in your eyes, this can occur in your GI, in your abdomen. This can occur any place you have a line going into or out of or something in your system. So a line being either a vascular line or a drain or a heart valve or anything else that's plastic or metal, prosthetic heart valves. You can get, this is a percutaneous line here. Pretty much any place you put a thing that's not in its natural state can form a biofilm. So this is tricky because biofilms are something that 30 years ago no one thought was a thing. Like everyone just said, oh, that's not what bacteria do. Bacteria grow in culture. They're planktonic. They float around. If what we've been doing is studying things floating around in a system for 100 years or more, they don't do the same things when they're floating around as when they're attached to the surface. So from a human health standpoint, we've sort of been studying the wrong thing. That's a problem because bacteria are not doing the same thing in biofilms as they do in other systems. And so really we're just at sort of the here be dragons sort of problem where we know that there's something going on, we know there's something wrong, but we have to sort of invent entirely new systems to sort out what's happening. So if I back up to here, this paper was a seminal paper that came out in 2007, I think it was. This was a big deal. So 12 years ago, we're like, hey, wow, biofilms are really important. That was 12 years ago. Microbiology's been a thing for a couple hundred years, and we've sort of only started to sort out what's happening in a biofilm state very recently. Okay, so why do we care? Well, I've already mentioned that there's an increased antibiotic resistance. This is not just because the biofilm is a physical structure. Most bacteria change about 10% of their genome when they go into a biofilm state. Some bugs are more than that. Group B strep is about 35% of their genome switches over. That's a huge amount to have transcriptional regulation at that level. If you had something that was 5% different, you'd call it a completely different bacterium. So antibiotic resistance is a problem, and what we've mostly done is sort of the shotgun approach, where we've said, here's a pharmaceutical compound that kills stuff, so let's just use that. That's like using artillery on something. That's a problem because we need something more selective than just throwing everything at it all at once. Why? Because most surfaces in your body have a microbiome. The classic one is to think about your GI tract, but that's true on your skin. There may even be a normal level in your bloodstream. There's some just evidence looking at that. And so there are places where you don't want to interrupt that, because if you do, you've now caused a new problem. This is sort of the classic example of this would be the C. diff problem. If you interrupt the normal GI flora, you cause a problem. Okay, before I talk about what we're actually doing, I want to emphasize this point. A lot of the things that are difficult about studying Enterococci are because they're incredibly tiny, okay? If this is sort of your normal neutrophil, about 10 microns, these are all to scale, and a yeast cell is kind of about half that diameter, so that's sort of a reasonable scale there. Okay, this is an E. coli, so it's a lot smaller That's Enterococcus. Okay? It's really, really small. And it's not just Enterococci. I mean, streptococci are really small too, but we often forget that. You can fit about 15 of these inside of an E. coli. You can fit about 4,000 of them inside of a mammalian cell. So a lot of the difficulties with even finding this organism are just that it's so small. It's basically like a really, really big virus. I mean, it's that small. Okay, so biofilm formation is a developmental thing. It's not here's a route, we go from A to B and then we're done. It keeps cycling around. So you start off with planktonic growth, you get an infection, it floats around in your bloodstream, and then it starts to interact with the surface. So first you get sort of this small amounts of interaction, and that's adherence, and that's reversible over the course of a few minutes to a few days, a few hours. And then you get further expression of surface proteins, and you get an actual attachment event. That's a little less reversible, but now it's firmly stuck to the surface. One of the things that's important to remember about biofilms is they are not just like transiently like, hey, we're kind of stuck to the surface. They are stuck to the surface. You can do bad, violent things and they will not come off, okay? So once that happens, then you start to form this extracellular matrix. Now remember, that's produced by the bugs themselves. Now you have a microcolony that's formed, okay? And then that grows and continues and becomes bigger. You get a mature biofilm, and then they start to come off, okay? And as they're coming off, we're now back in this planktonic phase. We're reseeding someplace else. This whole thing just keeps going around in a circle. So a lot of the work we did early on was looking at what happens in early stage biofilm formation. What happens in the first few hours? So this, by the way, this is 6 hours. So after 6 hours, these are individual enterococci. This is a scanning electron micrograph. They're stuck to a surface. So it's a piece of plastic in this case. And all this fuzzy stuff around them is the matrix that they made. And that's in 6 hours. So this is not a long-term sort of process. So in addition to that, we sort of characterized what else was going on. So the little green dots here are showing extracellular DNA, which turns out to be important in early biofilm formation. If you take that away, they fall apart. This is just showing the same thing, but using light microscopy. And they have a fairly complicated structure. I mean, this is not just little tiny things randomly stuck to the surface. They are associated with that surface. So really what I want to talk about today is the fact that Enterococci really has two entirely different roles in your body. As I said before, most of us have Enterococcus faecalis in our GI tract, and it's a normal commensal organism. In fact, Enterococcus faecalis is usually the first, and if it's not the first, it's the second bug that colonizes your GI tract after you're born. Within the first day or two, that's what's there first. Sometimes Clostridia beats it out, but usually it's Enterococci. And so that's the normal thing. It's supposed to be in your gut. That's where it lives. But then there's this whole role of Enterococcus faecalis as an opportunistic pathogen. There's this long-running debate, and every few months there's another article that says the opposite of what the one before said. When you have things like VRE infections, vancomycin-resistant Enterococci, in a clinical ward, there's two ways to think about it. Either one patient had it and spread it to everybody else, or everybody has their own and spreads it to themselves. And the evidence is all over the place as to what is actually happening. The answer is it's probably both. But regardless, there's this sort of dichotomy between how they exist when they're happy and how they exist when you're unhappy. So what makes that happen? How do we go from having everybody's happy to everybody's unhappy? And to do that, I'm going to talk about two different model systems I've been working on. One is the normal part, which is how do they colonize the GI tract in mice. We have a murine model we work with mostly at Mayo. And then the other part is what happens when you look in rabbits and how they cause endocarditis. So those are the two sort of stories I want to talk about today. So the first one, this is a model we work with down at folks at Mayo, mostly because they have a germ-free mouse facility. And there are lots of ways to talk about what's happening in the microbiome. You can look at a normal microbiome and how it's perturbed when you do antibiotic treatment to it or when you introduce a new bug. You can do all that. The GI tract is really complicated, and we really don't understand what's going on in a lot of levels. And so to get around that, what we decided to do was let's take a mouse that has never been colonized, was born and it would be a C-section, has never had any GI tract bugs, and start introducing bugs and see what happens. A reductionist approach, yes, but the alternative is let's try to figure out 1,000 variables all at once, which is not a good way to do science. So if you look at the GI tract in humans, we look at the whole thing, so going from the duodenum down to the colon. It used to be thought, and by used to I mean in the last 10 years, it used to be thought the small bowel really didn't have any bacteria in it. And in fact, we talked about small bowel overgrowth as being essentially any bacteria that were there. That's not true. There are bacteria that live in your small bowel, and they're supposed to be there. They don't live at nearly the same numbers they do in the colon. But in your duodenum and jejunum and ileum, you have anywhere between about 10 to the 1 to about 10 to the 7 CFUs per mil. Now again, that's very much less than 10 to the 11, 10 to the 12 per gram you get in the colon, but it's not 0. And there is a very different predominance as to what bacteria live where, depending on where you are. So you start off with sort of gram-positive cocci and rods, you get streptococci and things like that. And as you go down, then you start to get Enterococcus kind of in the jejunum, stretches into the ileum, to lactococci and some other bugs. And by the time you get to the colon, it's mostly anaerobes, okay? Bacteroides and things like that. So why? Why is that different? Well, we tend to think of the GI tract as it's the GI tract. That's sort of like saying the circulatory system. Not every part of your circulatory system is the same, and neither is your GI tract. Your small intestine does not do the same things, is not the same architecturally as your colon. GI tract is there basically to absorb nutrients, and your colon is essentially there to absorb water, if you want to separate it that way. But there's also very significant variations as you go down the GI tract as to what's going on. So the oxygen level changes as you go down. You go from having reasonably high oxygen levels to essentially none. The pH changes. It starts out very acidic, coming out of your stomach, obviously, and it's neutral-ish, 5.5, 6, by the time it comes out of your colon. What nutrients are available changes completely. So obviously, you're absorbing nutrients as you go down, so small-chain fatty acids are available in one place, not in the other. Also, the level of sort of immunological surveillance is important here because your GI tract is a huge mucosal surface that's involved in sort of sensing what's going on in the environment, and that becomes a big issue in this as well. And also, what other bugs are nearby you? So if you're trying to live in this area, you have different competition than if you're living in this area. Is there variation? Of course there's variation. And one of the things that's come out of a lot of the microbiome studies in the last few years and looking at normal humans is that this changes not on a weekly, monthly, yearly basis. This changes hourly. So what you have in your this is a dynamic process and things are moving around. Of course, that makes it harder to figure out what's going on because now we have a moving target. So as we go through the GI tract going down, like I said, pH is changing. It's going from acidic to neutral. The O2 is decreasing. It's becoming more anaerobic. The nutrient level and the flow rate is changing, and the bacterial load is generally increasing. So we're starting off 10 to the 1, 10 to the 12, and what's there is different. There's another complicating factor. Not only are you dealing with a long, skinny tube that you have to think about what's happening at one end versus the other, there's variation as you go, as you look at the tube from the end, there's variation from the epithelial surface to the middle, and it's not the same. And so what bugs are where in a radial direction changes. So if this is your gut epithelium here, you have a mucus layer that's different in the small versus large intestine, and where the bugs are changes. So Clostridia, Enterococci, Lactobacillus tend to be on the epithelial surface or the mucus layer. Things like Bacteroides, the Enterobacteriaceae, Streptococci tend to live more towards the lumen on top of the layer. So there's less accessibility when you're in this group directly to the surface, but there's also less host response because you're farther away from it. All that stuff matters. So this is what a mouse GI tract looks like. That's coming directly out of the stomach. This is the rectum down here. This is about 50-ish centimeters long, though even in mice that are all the same age, there's about a 30% variation from largest, and this is true in humans too. We have about a 30% variation in normal length. That makes things more complicated. It's not like they're all exactly the same size. One thing to note, so of course the small intestine, much bigger than the large intestine in terms of length, but much larger radially down here. So this is the cecum, and this is a mouse that has never seen bacteria before. This is a germ-free mouse. This cecum is incredibly enlarged, and this is normal in a mouse that's never seen bugs. A normal cecum would be about that big. If you take a mouse like this and you give them bacteria, it doesn't matter what they are, you just give them via gavage, it takes about 3 to 5 days and this shrinks down. And part of that is because your GI tract is involved very heavily in immune surveillance. And if you don't have any bugs to surveil, then you basically have a chronic inflammatory state. So your immune system ratchets it up saying, I can't see any bugs, I need to see bugs, and keeps ratcheting up what it's doing, trying to find them. Once you give it to them, that goes down. So the first section of stuff I'm going to show you is working on this sort of protocol. We took germ-free mice. We gave them Enterococcus faecalis, either just as a wild type or in some of these cases as a small pool of about 11 different strains. We kept them going for 7 days and then took the GI tract and sectioned the entire thing. Did this on about 20 or 30 different mice. 1 centimeter section, so about 50 sections per mouse, times 20 mice, that's a lot of sections. And these are not just block sections, but each of these, so every 4 centimeters you had a repeat of stuff going to SEM, stuff going for immunofluorescence, stuff getting put in the block, stuff getting banked for later. So we have this all the way down on these mice. So just to sort of orient you, this is the duodenum. This is the normal surface as it looks here. These have only been in for 7 days. They don't have almost any mucus layer. It takes about a month for a mucus layer to form. We can talk about that some more in a minute. So if we sort of look at this, these are scanning EMs. So this is leftover stool, retained stool. That's what that looks like. And then we have all these little things sort of sitting around in clusters. Everywhere there's a green arrow. Let's go look at those clusters a little bit more. So this is the normal surface here, little bits of junk here, and these are enterococci. How do I know? Well, that's the only thing we gave them. In addition to that, we've also checked to see, are they enterococci? Yes, they are enterococci. And these look pretty much just like the things that we saw when we looked in vitro. So that was nice. They are forming little microcolonies. And notice that they're not covering the entire surface here. So the standard model used to be for microbiome stuff, bacteria are covering the entire surface of your entire GI tract. That's not only wrong, but it's sort of ridiculous. Your cells have to absorb nutrients and absorb water. If there's bacteria covering every inch of them, they can't do that. So a normal microbiome can't be everything covered all the time. So let's keep going down. As we look at the duodenum and the ileum, we have these little clusters again, enterococci attached to the surface, in clusters forming this extracellular matrix. Okay, so that's reassuring. They're doing something similar in vivo as they were in vitro. And zooming in on them, so one of the tricks starts to come in here is that it's hard to take a really sharp picture at this level because we have to deal with both the host surface and the bacterial surface, and they charge differently. So that is a limitation I'm still sort of working on. Looking in the cecum is a very convoluted organ. It's kind of like looking at the bladder because it has to stretch a lot, and that's why it has a sort of rippled surface in here. And again, we get little clusters of bacteria, clusters here, clusters here. Now we're switching into the colon. The colon changes. The colon is no longer an absorptive for dealing with nutrients. It's absorptive for dealing with water. So we have these large folds going on in here, in like this. So if you look along the surface, you're like, okay, well, it's a piece of damage there, obviously. But so that looks like relatively normal surface. What do we have going on in here? As we zoom in on them, little clusters of stuff. And we have little clusters just like we had higher up. Now, one thing to notice here as well is that the surface of the epithelium is actually relatively happy. This is what normal epithelium looks like. Again, it doesn't have a mucus layer in this case, or at least not much of one, but there is not a grossly inflamed surface here. Now, I told you that these were in an inflammatory state because essentially we have bacteria, a bacteria-free state before we put these in. But it only takes a couple days for them to calm down. If you look at ones that we've never given bugs to, this surface looks unhappy, looks inflamed. Okay, so that's nice. They look like enterococci, and the PCR stuff says they're enterococci, but I'd like to know that they actually are enterococci, because we don't do science by saying things look like other things, they must be that thing. So also, as I said, we did paired sections, or quadded sections all the way down. So looking at the H&E. H&E, if we're looking at biofilms, is useless. Basically, no part of any biofilm stains with anything on H&E. This, by the way, is one of the reasons why I think it's taken us decades to notice this. Because when we do H&E sections, we don't ever see anything. And it doesn't matter what the biofilm is. You look in the gut, we basically wash it all away when we do the xylene processing. There are ways to help that, but we can't always just rely on H&E. So if you do a tissue gram stain instead, and notably it has to be a Huckert-Schwartz. If it's not, you don't get the same level of preservation. So that's the other part. Is even if you do a tissue gram stain, if you do a Brown and Bren, it doesn't work very well. And it has to do with how well the stains are penetrating the biofilm itself. So if you look at, these are the same sections from here. See these little clusters here? What are those things? Oh, those are little round cocci-sized things that are staining blue. Okay, so this looks like it's bacteria. That's nice. There's a lot of them, sort of stuck around in various sections, but it's not like they jump out at you. It's not like you're like, oh wow, what's going on? Well, remember, it's not the entire surface is covered. And until about 10 years ago, we thought the whole surface must be covered. Why do we not see the whole surface being covered? Because that's not how bacterial biofilms work. It's not how your microbiome works. Okay. So that's nice. Now we have things that look like gram-positive cocci. We have things like PCR that says they're gram-positive cocci. They look like the same structures. Now we're going to put in a bug that has expressing a fluorescence protein that has a marker that shouldn't be on anything else. We're going to put in a cyan fluorescent protein. There's nothing in your gut that's blue. Okay? Nothing fluoresces blue. So if you look at them in vitro, you can do CFP, YFP, paired staining. So that's what they look like in vitro. Nice staining there. This is what it looks like in the GI tract. So part of the problem with FPs, especially in the gut, is they don't last very long. You can't fix these and look at them next week. You have to look at them within a couple hours or they're done. So this is basically just staining for a couple of components on the villi here. What's this blue stuff? We blow that up. Little chunks of enterococci. Okay, good. So by SEM, by PCR, by staining, by fluorescence marking, they're enterococci. All right, I'm pretty convinced they're enterococci now. I don't have to worry about that too much. Okay, that's good. So what's sort of the take home from these sorts of studies? Well, the first thing is it's the same all the way down. Now, how long does it stay the same? We followed these out to about 35, I can't remember, 35 or 42 days. They're the same. If you don't put any in any competition, they're just enterococci all the time. And they look like this pretty much all the time. They don't overgrow, they don't cover the surface. Mice don't get sick, they're happy. Well, I'm assuming they're happy. I haven't actually asked them panels of questions to determine if they're happy, but they seem happy as mice get. Now this changes if you start putting in other bugs. If you give them competition, that changes. But overall, they appear to not really care where they are in the absence of any competition. So you have these discrete little colonies, okay? They look like the stuff we see in vitro. Do I know that they're biochemically exactly the same? No, haven't done that stuff yet, but they look the same. That's actually old data. They look the same over 35 days. The microcolonies stay the same size, roughly, from a few dozen to a few hundred cells. That's interesting. Why? Why wouldn't they just, we generally think about bacteria as like, okay, well, they would just grow to fill up the space. They don't. So obviously there's either something happening between the microcolonies to tell each other, hey, this is as much as we should grow, or the host is telling them they shouldn't grow anymore, which would also be interesting. And there's not really any gross inflammation going on. Okay, that's good to know. That's great, but germ-free mice are also very weird. So I've just told you they have a disrupted immunological response because they haven't had any bugs there. So what do we get when we start looking at things that are more normal? Because it's great to look in this restricted system, but we should check to see how that actually works. So we did a small set of experiments looking at mice that had normal mouse, at least normal lab mouse GI tract. So these are specific pathogen-free. They're not known to have any pathogens for mice, but otherwise they're just sort of normal bulb C mice. So as you look at them, first of all, these are mice had a normal microbiome. We gavaged them with some enterococci. We waited a week, sacked them again. So overall, the same thing we saw before, normal microbiome, normal surface stuff here. This is actually some retained mucus over here. That's what mucus looks like. Mucus is actually really hard to study. And this sounds sort of silly. It's like mucus, that's gross. Why would we want to study that? It turns out to be really important in your GI tract. And not only is it really important, but it's really important to understand the structure of what's going on in the GI tract. There's about 3 labs in the country that do this, that study the GI tract mucus at all, because mucus, we think of it just as being sort of unformed blobs of polysaccharides. Well, unformed blobs of polysaccharides are hard to stabilize. They're hard to figure out what's going on with them. This is a tricky thing to study, and this is why most people just blow it off and say like, I'm sure it's fine. But I think it matters. I think it actually matters because the mucus layers are actually different in your colon versus your small intestine. In your colon, there's actually 2 separate mucus layers that interact with each other. So it's not just epithelium, mucus, bugs. It's epithelium, mucus that's one way, mucus that's another way. And it has to do with what the saturation of that is. So as we look down there, we can see some nice immune surveillance sort of stuff going on, little pockets of cells. Okay, that's great. Well, this is a little different. So now we have these little microcolonies, but these are not all enterococci. Some of these are enterococci, but some of them are just normal microbiome that you get from a mouse. As you start to zoom in on it, so this is now a little bit of retained mucus in there, everything sort of looks fuzzy. We start to get these pockets of things that kind of look like the things we saw before. Hey, look, there's enterococci. Again, I'm not showing it here, but I'm not just saying these are enterococci because they look like it, even though they do. But we can tell you that there are enterococci in there, and this is what enterococci look like. And in this case, they're not directly on the surface because they came in later. But again, even when it's not just enterococci, there are these little clusters all over the place. There's some work being done down at UT. Marv Whiteley's working on trying to figure out why this happens, because it turns out there are very distinct spatial orientations depending on what the bugs are and sort of how friendly they are to one another. And so if you put bugs that are friendly to one another, they grow closer than ones that aren't. Is this a competition thing? Is this a germ warfare, so to speak, thing? Don't know. But this is an active area of research trying to sort out kind of what's actually going on. And so, these are little clusters of enterococci that look just like the things we saw before. Some of these things that are over here are bigger. They're bacilli. They're not enterococci. But we'd expect to see them in sort of a normal GI tract. Interestingly, we also see these long, funny, stringy things. I I don't have a picture of it right here, but at the beginning I showed you some pictures that had long, stringy things between them. These are probably extracellular DNA. That's what some Enterococci formed biofilms early on, and these look exactly like those. I haven't proven that yet, though. Okay, so if we look at sort of germ-free mice versus wild-type mice, they're not exactly the same. There's clearly some differences, but they still form microcolonies in both cases. It's a little tricky to sort of sort out how one can answer the question of why is that different. We've done some studies where we take the Enterococci, put them in first, let them colonize, then add a normal microbiome after it. We've done the reverse. That's still an area we have to sort of sort out a little bit. But overall, one thing you can say is that in neither case do they look inflamed and unhappy. I mean, this is the normal state. After all, you'd sort of expect that. And this spatial relationship between microcolonies is probably important. Have to sort of sort that out. Okay, so then we decided to do a small study to sort of sort out, okay, what happens when you look at different mutants? What genes are important in colonization as a first step towards figuring out what genes are important in pathogenesis? So this is a small pool of 11 different strains, some of which we know some things about, some of which we don't. And we did a paired run where we put them in our bioreactor system, just an in vitro system on the bench, and then also put them in the mice. And the first thing to note is that these ranking numbers are different. Well, that's good because mice are not the same as plastic and glass. So if they were the same, I'd be really nervous. But overall, what we can say is that there don't tend to be things like things that are high here are completely low here. Now there are a few, 2 and 3 kind of go down here. And some of these, like I said, we don't know much about. But the goal here was simply to say what happens when we look at a few genes, do we get a difference? That we do, okay. So in Gary's lab, they just finished publishing a study looking at, okay, let's now do the first set of this chunk where we look at a much larger set of genes and we just look at the in vitro system first. So we'll go into a mouse later. But this is looking at a pooled transposon library array covering about 75% of the entire genome. It's actually practically a little higher than that for sort of complicated reasons, but we'll stick with that number for now. And as the biological system, we're sort of looking at bile resistance in our sort of standard lab strain. It's about 7,000-ish mutants looking overall. Now, this is one of the classic genomics slides you get, which is that I can't read that, and I don't expect you to. But essentially what we can say is lots of genes are up and down regulated. Okay, that's great. Who cares? Well, who cares is let's figure out what this means when we can look at a few of these genes and sort out, does this make sense from a bioresistant standpoint? So picking 4 genes that we know a little bit about, some more than others, just looking at growth curves. And what we can say is that, okay, we look at DNA-K mutants, they behave the same way when we look at traditional techniques, plating techniques versus genomic techniques. Okay, sanity check, that's good. When we look at cell wall signaling mutants, they behave the same way, let's say, same way as well. Okay, that's good. And then we have 2 hypotheticals here that we don't really know much about, and we can also get the same data in vitro about them as well. Okay, that's excellent. Now we're going to do this in mice. So that's the next step is, of course, we know that they should be different in mice versus in vitro. Now that we've done the sanity checks, we can actually move on and do that part. Okay, so that's the GI tract stuff. I've left out a lot of stuff just for time, but I want to have time to get to this sort of second large system, which is looking at an endocarditis model, again with Enterococcus faecalis, trying to sort out what's going on there. So, completely different model system, looking at a different organ, and the results are somewhat similar, which is interesting. So this is sort of your standard med school summary for endocarditis. Endocarditis has to do with the endocardium. It's inflammation. You have some bacteria. You have an abnormal substrate. You have bacteria in the bloodstream. You get thrombi. Boom, endocarditis. As with most things in med school, half the stuff on here is actually wrong, but most of it is, at least, they're trying to be correct. And this was from Tulane. I'm not picking on Tulane. Every med school wiki has something like this. But the important part is, you have something going on with the endothelial cells in your circulatory system. All right, there's a heart. I'm going to talk about stuff as being from the right side, from the left side, at the aortic root, and at the arch, because that's sort of where we surveyed it. So this is an old system, old in that people have been doing this for a long time. And the model of system essentially is, you take a rabbit, you incise in the left carotid, you put a catheter in place, and you slide it down until it starts bumping into the valve. So you're physically, mechanically causing damage, so you can create a space for endocarditis to happen. So it did that. After 2 hours, you remove it, close up the incision, inject about 2 × 10⁹ bacteria per mil into the ear. That's a pretty decent dose, but it's not insane. I mean, this is kind of what we would normally do. Goes for 4 days, sacrifice the animals, and go look at the heart for vegetations. So a previous postdoc in the lab who is now working out at Uniform Services in Bethesda was doing this for several years, and we sort of had this problem, which is that, so generally speaking, so if you did this with staph or strep, they would all be dead. Okay, staph and strep have a very aggressive course with endocarditis, and they just kill you. Bugs like Enterococci do a subacute bacterial endocarditis, and what you get are symptoms that just sort of drag on. So, and somebody had subacute bacterial endocarditis, showed up in the clinic, they would be the one that's like, I just don't feel myself, I feel kind of crummy, I'm tired all the time, and you do an anemia workup, and then you'd be, come back in a few months, we'll see how you're doing. And they come back in a few months, and then they don't have any heart valve left because it's gotten eaten up by the bacteria. But this is the sort of thing where with Enterococci, they're pretty much fine after a few days. Not really sick. But what would happen is that we would pull these out, and sometimes we would have lots of bugs on the valve, and sometimes we'd have no bugs on the valve, and that didn't really seem to have any relationship to how many bugs we could isolate from the bloodstream, so whether they were bacteremic or not. And Christy spent a lot of time trying to sort out what was going on and what was wrong with this. And it was tricky because as you're doing all these steps, there's a bunch of places you could imagine something going wrong, okay? But really, if you're putting the same amount into each animal, you should be getting roughly the same amount out. I mean, yes, they have different immune responses, but it shouldn't be going to zero. So that we struggled with this off and on for a few years, because sometimes it would work great for a while, we'd have lots of valvular colonization, and then sometimes we wouldn't. And so we decided that we got kind of desperate at one point and just said, all right, let's just look at the rest of the heart. Let's see what else is going on, just because maybe the Enterococci are someplace else. Enterococci do weird things. So we took the catheterized animals and we looked in the right heart, and this is not the valves. So standard endocarditis, when people say endocarditis, what they mean is valvular endocarditis. Nobody ever just talks about endocarditis being anywhere else. But that's not what the word means. The word means colonization of the endocardium somewhere. But that's not what people say. They don't say what they actually mean. So this is in the right heart. Okay, this is nowhere near where we've actually poked the animals, and yet we have these little clusters that look exactly like the stuff I showed you in the gut. If you look in the left heart, you got these big clusters. I mean, this is endocardium. This is a vegetation growing out of the endocardium. This is not the valve. This is just in the left heart. That's interesting. Okay, so the aortic root, lots of bugs. Aortic arch, lots of bugs. So now we have something all the way from the right side, the left side, all the way through to, so there's no reason this shouldn't be systemic. I'll just cut to the chase. This is systemic. But this in the catheterized animals, we're like, well, that's weird because catheterized should only matter at this point and maybe this point if you were not very careful and you pushed all the way through the valve. But how are you getting stuff here and here? Because the standard model requires that you cause damage. That's why we're putting the catheter in the first place, because you have to damage the surface. Okay, that's weird. This doesn't show up very well in here, but so this is actually just a piece of aortic root out of one of these rabbits. There's a lot of backbone fluorescence here, but at one point we got really desperate and just said, let's just label the bugs before we even fix anything. We're going to put in an analog, a vancomycin analog that's fluorescent, and we're just going to go look to see where are they. And there are these little stringy things and these little clusters of green, and these are vancomycin. These are Enterococci that have taken up the vancomycin in about 10 minutes, and that's what they normally do. And this is the same field as this. So the valve has already been removed. There's no valve here, and there's all these little chunks of Enterococci everywhere. Remember, this rabbit is not sick, okay? Looks for all the world like a regular rabbit. And these little clumps and these little stringy things, and we normally talk about these, this would be on a valve, right? That's where they should be. That's where everybody says they're supposed to be. All right, so it's catheterized stuff, okay. So then we said, all right, well, what happens if we do the same experiment, we don't catheterize them? We do all the same stuff we did before, we just don't put the catheter in and cause any damage. And it looks exactly the same. Left side, right side, everywhere else. Okay, well, that's weird. All right, let's follow them out further. 14 days. Let's follow them out further and anesthetize them, but don't even cause any, like, we don't open them up at all. We don't do any incision. We just put them to sleep for 2 hours, wake them up, squirt stuff in there. Looks exactly the same. So this sort of starts to ask the question, at least in the rabbit model, do you actually have to have damage to cause an infection? Because again, The standard model is you cause damage. And I mean, usually subacute bacterial endocarditis, one of the places you see it is in people between about 60 or 70 and older. And the assumption is they've got calcified valves and that's caused the damage. But the assumption there is still there has to be damage. This would suggest you don't need any damage, okay? So if we do surgery and do catheterization, we get attachment. Yeah, we get that, okay. If we do that but we don't catheterize them, do we get attachment? Yes. Okay, so maybe no damage. What if we just do anesthesia? Do we still get attachment? We do. What happens if we just mildly sedate them? They don't even ever go under, because now we're worried, like, is there some inflammatory condition that's going on here that's being caused by the anesthesia? Doesn't matter. If you just take these and you basically give them what amounts to a rabbit benzo for 2 hours, they're fine. It turns out for 15 minutes you give them a rabbit benzo and they're fine. They still cause this. It still looks exactly the same. Followed out for 14 days, still looks exactly the same. Take rabbits you've never, for the obvious one, you take rabbits that you've never injected anything in, do you get those? No. Okay, good. The negative control works. That's good. So this was a very large review, came out about 4 or 5 years ago, that sort of talked about Nature Reviews Cardiology. So they re-emphasized this is what the standard model is, okay? You have to have an inflamed surface, you have to have damage, then you get colonization of the surface, then you get maturation, then you get detachment and dissemination. The standard model actually has something even more sort of complex in there, which is that it says you must have platelets and fibrin deposited before the bacteria can attach. That's not true, or at least not in the rabbit model. You don't need that step first. Okay. One of the things that's I don't have time to show you here is that one of the other things you would expect to have in any sort of endovascular colonization is if you have an immune response at all, you should definitely have an innate immune response to having a large amount of bacteria squirted into your bloodstream, right? One of the things that's notable about these images is that there are essentially no macrophages and any sort of neutrophil response at all. I've seen like 2 ever. Right? I see lots of little red blood cells. I see a few platelets scattered around. There's no neutrophil response. So as it turns out, and we've actually known this for a while, and I should have thought of it earlier, one of the things that biofilms do for bacteria is they make it so it's really hard to be opsonized and taken up. And that's because there's a size limitation for macrophages. Once you get bigger than about 25 microns, you can't be engulfed anymore. Microcolonies, these bacterial microcolonies are much bigger than 25 microns. So essentially, you are completely hidden from the macrophage response. Okay, so this is what things look like in vitro. This is what it looks like after a few hours on a surface. We've talked about, we can label the properties of the ECM. There's some cryo stuff that we did. So in rabbits, in the endocarditis model, and in mice in a GI tract model, they all look pretty much the same. One of the things this suggests is that this biofilm state for these bacteria is a very deeply conserved property. This is what they want to do. This is what they want to do on pretty much any surface, no matter where you are. So they're all pretty much the same. In gross morphology, anything from time ranges from a couple of hours to 35 days. Biofilms form microcolonies, stay that way pretty much the whole time. What I haven't shown you, we've also done this on a variety of different surfaces on plastics and biopolymers. We've also done this in porcine cardiac valves that have been explanted, and then we infect those and look at those. There's a rat osteo model we worked on with Mayo for a while. They all look just like this. Okay? So one of the things that often comes up at this point is people say, okay, great, why did we never notice this? Okay? Talk briefly about it with the H&E stuff. So biofilms turn out to be, biofilms are tricky because they're about 98% water. Okay? That means it's really hard to preserve them. So there was a faculty member at the university about 15 years or so ago, Stan Erlandson, who did a bunch of early work on this with Gary, looking at how do you preserve this. And it turns out if you add what we all know in the room is a very standard extracellular matrix dye, if you add alcium blue, it holds the matrix together during processing. And one of the things I can tell you is, because one of the things that people ask is, well, how do you know the alcium blue isn't causing this and making it look like this? So this is actually cryo-EM that I did looking at, so this is where there's no alcium blue. Everything looks fuzzier because it's cryo, but you get the same structures, the same things form. So it's not the alcium blue doing something, but if without the alcium blue, none of this stays around. And if the matrix doesn't stay around, all the bugs fall off. So I think what's happened when people have looked at EM over the last several decades looking at endocarditis, because they didn't actually preserve the bacterial extracellular matrix, what they saw was essentially a skeleton. What they saw was, here's the fibrin, here's the platelets, that all stayed together on the surface of the damage, but all the bugs went away, so I didn't see anything, so I didn't talk about it. I think that's what happened. Okay, so what's sort of the 30,000-foot view? This is an ex vivo model I've been working on as well. This is so it's great to do this stuff in animals, it's great to do this stuff in vitro, but it'd be nice to do something that's halfway in between. And so this is a CACO2 model. These are Enterococci in little clusters growing on the surface of an ex vivo model for that. So the 30,000-foot questions for all this stuff are, how is colonization and development actually happening in the GI tract? So when we started looking at this a few years ago, this is all stuff we sort of assumed everybody knew. And it turns out no one has done this stuff since the early 70s to look at colonization. Mostly because it's hard. It's really hard to sort of sort out what's going on when you have all of these different, when you have an entire human system and an entire bacterial system and an entire bacterial microbiome on top of it. So to sort of sort out what's actually happening, what are the developmental events, how is that changed by what happens in the normal model? We've got this TNC library that we're working with now. What genes are actually essential in vivo for attachment, and also in vivo for then eventually pathogenesis? Because first you have to sort out the normal state, then you can sort out what's actually going on. There's other folks in the lab also working on the conjugation model, and they've been able to demonstrate that conjugation, or horizontal gene transfer, happens in the gut, and it happens very efficiently. But it actually matters who goes in first. It matters whether or not the recipients are there first or the donors are there first, which has some intriguing other possibilities attached to it. But this is, of course, a major way that horizontal gene transfers happens for antibiotic resistance and other things. We have a model as well working with folks out at Mayo on a humanized microbiome. So essentially this is mice that have a human microbiome. So you take stuff from the donor, from a donor human, gets put into a mouse. And we looked at that a little bit as well. And also just the normal system. We've been talking to Dave Massipus a little bit about this. Lots of stuff that we do in the lab, especially for microbiomes, has really been hamstrung by the fact that we all like to look at really well-defined systems. And that's great, but that's not reality. The reality is things don't work that way. If you take a microbiome and you try to say, let's just use 4 bacteria or 12 bacteria, 16 bacteria to act like that normal microbiome, that's not the same as 500 or 1,000, which is what's normally there. So we have to sort of move into that stuff as well. From the endocarditis model stuff as well. So originally when I did these 2 models, started them in my postdoc, my original idea was, well, one of these will probably work. They both worked, which is great, but also makes things more complicated because now I have 2 sets of things I gotta make sure we get to. I gotta work out, is this damage requirement really true? So the evidence that we have, and this is now over about 35 or 40-ish animals and about 15 different experiments, I have not been able to find that endothelial damage requirement exists, at least in rabbits. So we've also got to figure out, and we've talked to some folks in St. Paul about this, is this true in, say, pigs? We've got to figure out another animal model to make sure this is not. I mean, rabbits are kind of weird, and they have kind of a weird immune response. Sort of their anatomy is a little bit odd, so I don't think that's the problem, but I got to sort of make sure that that's not the thing that's causing all of this. Is there a host strain variation? Probably. Like I said, the porcine stuff. We do run into problems occasionally with the animal use committee because they see us injecting stuff into their ears and say, oh my God, you're making the animal septic. And it's like, well, yeah, but they're not sick. But they don't want to let us go longer time periods, but we're working on that. So, for both of these, there becomes sort of this question of why is there so modest an inflammatory response? So if you look at the endothelium is just as happy as the GI tract was. That's weird. You have a lot of bugs floating around in your system. So this turns out to be both complicated and interesting. There was a South Korean group about 10 years ago who found that when Enterococci go into a biofilm, they actually start producing gel E, which is a compound that's involved in sort of remodeling of the biofilm itself. And this turns out also to cleave C3 in complement. So if you're not able to be engulfed and you're bypassing the entire innate immune response by interfering with the cascade, now all you have is an adaptive response. And if you don't have anything immunogenic on your surface, how do you actually mount it? So it becomes sort of really complicated to figure out how would you actually mount a response to this. In addition, Kim Klein, who's now at Singapore, has been looking at this and has found recently, it's not published yet, that if you look globally at markers of inflammatory response and look at sort of the big 10 or 12 that you'd expect to see, before you put Enterococci in a germ-free mouse, everything's sort of off the charts. The inflammatory response is huge. Put Enterococci in, within 48 hours, they all come down. Put a different bug in, doesn't do that. So Enterococci are doing something to knock this immune response down, which in a larger sense sort of makes sense if you think about the fact that if they're one of the first bugs in our GI tract, that's one of their first things they have to do to stay there. Okay. The sort of larger question of how does immune surveillance work before there's anything there to surveil? If there's no bugs there, how is this actually happening? A lot of people are working on that. Not a lot of progress is being made at this point. And we do have to keep in mind too that we've sort of been assuming that mice that have never had a GI tract microbiome are basically like neonatal mice, but that is an assumption. So we got to check that to make sure that their immune tolerance is actually working the same way and it's not just an artifact of the system itself. So with that, I'd like to thank obviously Gary for all the support over the years and letting me continue to hang out over there. Christy Frank, who's now at Uniform Services, has a faculty position out there. We also work with the Patel lab down at Mayo, and as well as Perna Kashyap's lab. He has the germ-free mouse facility as a gastroenterology. I'd like to thank Dr. Ferreri, and then that's the funding sources for most of this work and some references, and I'd be happy to take any questions. Dr. Trussell.
Aaron, thank you very much. That
Sure.
was an absolutely fascinating talk.
Oh, thanks.
I want to make a comment and then ask you a question. Obviously, not all endothelial cells are made the same. And obviously, endothelial cells and epithelial cells in the GI tract are much different.
Yep.
And the question I want to ask is, do either the endothelial cells somewhere in the body or the epithelial cells contribute to the formation of the biofilm, or is it all bacteria?
That is an outstanding question and one that I have been trying to figure out how to answer for quite a while, because the fact that the biofilms, at least on a gross level, look the same on both surfaces was a shock. I did not expect that at all.
Yeah.
I expected them to be quite different because they're different surfaces.
Yeah.
Which is one of the things that sort of led us to thinking about, if this is the same on plastic and the same on epithelium and the same on endothelium, this must be a thing that they just do. And to be fair, most bacteria spend most of their life in the biofilm. That's part of the reason why this is tricky, because we study when they're floating around in the microlab, but that's not what they really do. A lot of the surface attachment factors for Enterococci are not well known. It has been known that for a long time that there's a von Willebrand factor receptor, but of course that you should only have that when you actually have endothelial damage.
Right.
That shouldn't be just a thing that's sitting out there. But it has not been worked out as to what the attachment factors are, and in fact, even in things like staph and strep, it's not always clear how they work in vivo. In vitro, we can make them work sort of one way, but we may be hijacking kind of what they're doing. Things like group B strep, it's a little better understood. Staph aureus is a little bit better understood. But even there, typically what we do is we know one or two that work really well and we skip the rest of them.
So in your model where you inject the bacteria into the ear vein, is the localization on the cardiac endothelial cells based on the way that you injected them, or is it due to the fact that the endothelial cells in the endocardium are different from endothelial cells in other parts of the body?
So we've looked at a couple of places elsewhere in the body, looked at highly vascular places like the kidney and other things.
Yeah.
We've only done a couple of them. We have a lot more in blocks. They appear to be the same. So one of the things that's always been tricky about endocarditis is if the place where you attach and you cause disease is in the valve, how does that happen? Okay, your valve is doing this.
Yeah.
Okay, that's not a really nice place for bacteria to land and just do stuff. Your endothelium is doing this.
Yeah.
Okay, and your endothelium in the rest of your body is pulsating less than that. So I suspect, I don't have data for this yet, but I suspect what happens is that the endothelium gets colonized, and it doesn't matter whether it's cardiac endothelium or elsewhere. That gets colonized, and then eventually over time you seed the valve. That also, from immunological standpoint, is very interesting too, because people have always suggested. So the inner 2/3 of your valves essentially have no immune surveillance because they're not vascular, no direct immune surveillance. So the idea has been, well, biofilms are protective because you can't get to it, you can't see what's going on. Well, if you're seeding someplace else, you have to now answer the question, why aren't they being seen? What's not happening here? But no, that's a great question, and I still don't know the answer. What's going on there? So, yes.
In your GI tract experiments, have you tried the effect of various diets?
We have not. So Enterococci will eat pretty much anything. They're pretty adaptable that way. There are folks who have looked at, so obviously your microbiome changes based on what your diets are. we haven't yet gotten to the point of thinking about what else is happening in the colonized system on that sort of micro layer yet. But it will definitely change. I mean, you can see in lab animals and in humans, you change the diet, within a few hours things start to change, and within a few weeks they're completely different. You go from vegetarian to not, for example. Dr. Firk?
Yeah, very nice talk there.
Thank you.
Very exciting. I'm sure this has been done, but is there any association or interesting information on E. faecalis and any inflammatory bowel diseases?
So there does not appear to be a direct connection to inflammatory bowel. We are working on a project with Dr. Rashidi looking at other inflammatory conditions, so in his case, graft versus host. And it turns out some of the other Enterococci may have some interesting things going on. So Enterococcus faecalis and faecium have been sort of the ones people have studied for forever. Faecium because it picks up VRE, faecalis because it picks up everything else. But there's another 16 or 17 Enterococci that exist, and they've been very poorly studied. But it turns out that bugs like Cassiflavus and Gallinarum and stuff may have a protective effect, and we don't know why yet, but may have a protective effect against GVHD. But the larger answer to your question is nobody really knows. Mark Hoyk at Oklahoma has been working on this sort of large scale, is there an association between colon cancer and Enterococci in the colon? That appears to be true, but the mechanisms for that are very poorly understood. So,
A follow-up question on
Yep.
The work that's been done by the people here in the C. diff patients,
Mhm.
fecal cancer, whatever. How does E. faecalis fit?
So that's an excellent question. One of the things that's really tricky to understand about how the fecal transplants are working is, are we actually recapitulating what happens when it's a native nothing is there state, or are we simply putting a Band-Aid on top of it and just saying, well, these work in another person, so they'll settle out and everything will just sort of be fine. And nobody really knows. It's one of those things where a lot of it is going to depend on how sort of scorched earth the GI tract is. If you're down to no mucus, no anything else, it's been that way for a while, probably it looks more like a neonate. If it's just disrupted, that's really hard to, so Enterococci do these really weird things when you get inflammatory processes going on. There was some work done here 20 years ago where people were looking at what happens when you just take a regular animal and sort of give it SDS, whatever, make it an inflamed state, what happens to Enterococci? And to everybody's surprise, it turns out some of them just translocate and go into the bloodstream. That's really weird. Most bugs don't do that. But Enterococci are definitely doing something, but we don't know what it is yet for the colonization part. So, yes,
Could I make a comment?
yes, absolutely.
There's some information that it's happened after Aaron left the lab, so you might not be aware of it, but I've been approached by a couple of different groups. One who works on C. diff, and it's a group at Vanderbilt, and they noticed that C. diff patients, when you just do culture, they also, their Enterococci go way, way up. Not necessarily, and so they've actually found that when you co-culture Enterococci and C. diff in the lab, the sort of global phenotype of the C. diff totally changes. So this group approached me because we have all these mutants, and they are now trying to find out what the interaction is. We have a project in my lab that a postdoc who was working on enterohemorrhagic E. coli in Texas. I was fortunate to get a free postdoc because her husband got a job here, and she's studying EHAC, enterohemorrhagic E. coli, and it turns out that co-culture of Enterococci with EHAC also increases expression of virulence factors of EHAC. So this might be something, my pet hypothesis is that Enterococci always do better in terms of population during dysbiosis. And you could think of, you think of antibiotic-induced dysbiosis, and Enterococci are there because everything else gets killed off. But of course, I would call EHEC infections or C. diff infections dysbiosis. And nobody has actually looked at the enterococci because they're not the cause. But this may be another mechanism that enterococci have to overgrow in the, so this is kind of a, this is all hand waving right now, but I think there's another story about the ecology of enterococci in these other situations. So.
Yep.
So you showed some of the images in the tissue colonization. There was some type of order for that. Where they would sort of nucleate in certain places and then grow in other places. There was some sort of order. Have you applied any sort of nucleation and growth type thing? So there's nucleation and growth models, typically in chemistry.
Yep.
But has that been done in bacterial work? Because it kind of looks like a nucleation and growth phenomenon in this, and there's probably likely some intrinsic thing happening in endothelium that's causing this to happen.
So we actually, depending on the strain you're using and everything, you actually can get something like this that's not even on a surface necessarily. You can have biofilms that essentially form in little blobs in culture. And that's because as the cells start to do things like conjugation, they get sticky. We have done some modeling work with the folks over in chemistry, but it's been more looking at the flow of information essentially that happens when you have donors and recipients and are exchanging genes. I haven't thought about it in terms of a nucleation concept by itself. I'll think about that. Yeah. So, yeah.
Thank you so much.
Thanks.